Great. Yeah, thanks for the introduction. I'm Konstantinos, and I'm working at DTU Bioengineering at the Protein Systems Biology group under the supervision of Professor Ulrich Aften-Keller. And here you can see where we're located. I mean, this photo is old, so that's why all the lights are on. But I'm here to talk to you about an assay we developed with high throughput and high sensitivity to monitor biomarkers in wound exudates. So I'll just have I just have a small background on wound healing, which can be divided into 4 distinct but overlapping phases. First of all, upon tissue injury, the hemostasis pathway is initiated with a coordinated response by coagulation factors, platelets, and other proteins to establish the fibrin clot, restore the barrier function between the organism and the environment, and prevent hemorrhage. Here you can see how it actually looks in the skin. Next up, we have the inflammatory stage, where immune cells and other cells are recruited to the wound site, and they express cytokines, and they begin the processes of pathogens, pathogen clearance, and phagocytosis. Next up, we have the proliferative states, where fibroblasts and keratinocytes migrate to the wound site. They profusely express extracellular matrix proteins, and they cover the site by proliferation. And finally, we have the remodeling phase, which is the lengthiest of them all, where collagen fibrils and other ECM proteins are organized and scar tissue is formed. Now, proteases are involved in all of these stages, and being a protease group, we would like to know what these proteases do and what are the substrates in the process of wound healing. And this is not only a basic research question. A dysregulation of protease enzymes and their inhibitors can cause aberrant physiology, which can result in chronic wound phenotypes. And these are a huge societal and economical burden, with more and more shares of the population being affected over time and in the next decades. So we'd like to know more about these processes, mainly proteases as well as other proteins, and what can we do to, of course, provide better treatment and have better prognosis of these wounds. So to achieve that, we partnered with a German wound dressing company, Hartmann, and we established a clinical study with a cohort of 43 impaired patients. So these are patients with chronic wounds, and 8 acute patients. So these are patients that underwent split thickness skin graft surgery and serve as a control group. And what we get out of this group is the bandages that were used to treat these patients over the course of wound healing. So we obtain 7 bandages in the case of impaired healing and 8 wound dressings in the case of the control group. And these are 2-week intervals in this group and 2-day intervals in the acute group. So we have time-resolved wound dressings or wound exudates from these patients. And in these wound dressings, we can have the wound fluid, which it serves as a signature, as a proton that we can use to pick up signatures of wound healing, because this wound fluid travels through the underlying tissues, and it has all these biological signatures that we can monitor. So after developing a protocol to extract these proteins, and we optimized this protocol, we stored them, and we continued with 2 different workflows. One was for biomarker profiling, because studies from our group have shown that we can have biomarkers for accurate progression and for accurate detection of progression of wound healing, and TELS and terminomics, which assesses proteolytic substrates. So for this biomarker assay, we wanted to monitor a set of proteins, 9 in total, that were involved in different stages of wound healing. So we have a complete picture of what's going on we decided to monitor collagen and fibronectin as extracellular matrix proteins, a few proteases that are involved in wound healing, as well as inflammation markers such as TNF-alpha, interleukin-1 beta, and calprotectin. And we had a few considerations in mind. First of all, some of them, especially TNF-alpha, interleukin-1 beta, are very low abundant in our proteomes. These proteomes are very complex. They're plasma-like samples, and they span a large dynamic range over 6 orders of magnitude. As you can see here, we quantified them with a multiplex ELISA, Luminex, and we can actually see these concentrations, and they span a quite big order of magnitude. We also wanted to monitor these proteins without any prior enrichment, as this would significantly add to the sample preparation, and we wanted, of course, to achieve high throughput and have an assay that we can process a lot of samples in a small time, as we had a large number of patients as well as time points for each individual patient. So for that, we used a SureQuant method, which is a bit different than the standard reaction monitoring workflows for targeted proteomics, in the sense that it relies on heavily isotopically labeled peptides to trigger the MS2 scans for our endogenous peptides. So the way it works is that we spike the heavy peptides in the mix, as you would do for the standard PRMs. But instead of having scheduling windows for the peptides, we acquire a low resolution MS2 scan that doesn't waste that much time, that much instrument time. And when we see enough transitions for the heavy peptides, so we're confident that the heavy peptide is actually eluting from the column, then and only then we trigger a high quality MS2 scan for our target, for the endogenous peptides. And then we, as we would with a standard target proteomics, acquire points across the peak, and we can quantify based on the duration of the area under the curve. So this method eliminates the need for scheduling windows, and since we're only acquiring MS2 scans for our endogenous peptides when the heavy peptides are eluting, we can use shorter gradients and therefore achieve higher throughput. So to establish our assay, we had to, of course, select heavy peptides, and we developed a tool to query available data from databases like Peptide Atlas and ProteomicsDB to find the most prototypic and have reference peptides for each protein of interest. We conducted survey runs to figure out which transitions we should monitor and whether our peptides fly well in with our setup. And we had to, of course, optimize our transitions, remove any interfering ions, and make sure that everything looks good. So after having the list ready with the peptides of interest that I would like to monitor, then we wanted to look at the gradients, how high we can go in throughput and how low, respectively, we can go in gradient length. And we optimized with 4 different gradients: 40 SPD, 60 SPD, 100, and 200 SPD. We had the best results with 60 and 100 SPD, and we can, of course, obtain more points across the peak with a 21-minute gradient. However, we didn't see any drop-off in sensitivity or any trade-off for that matter with 100 SPD, and since we can achieve higher throughput, we went with the 100 SPD method for the final assay. So here I'm just showing the final workflow after extracting the proteome from the wound dressings. We digest the samples, we spike the heavy peptides, we load directly on Evotips without prior desalting, and then we analyze with an Evosep One and an Exploris 480 setup. And with that, we were able to analyze 365 samples, our full data set, in less than 4 days with very high reproducibility and efficiency. We were able to analyze them non-stop without any interruption. And of course, we had a very complete data set. We were able to acquire data for most of the samples and most of our peptides. Here you can see the curated peaks. So these are actual peaks. There's not noise level. So everything that's noise level, we remove them. And I'm just showing a subset of the data, 6 patients, but we were able to have very high data completeness. So here I'm just showing a summary of the results in a gist. You can see the profiles across time points for the different biomarkers of our panel. And I'll just talk about a bit about the biology. We can see that, for example, we have high collagen levels and high fibronectin levels, at least not deposited proteins. And we can also see that for our inflammation markers, for example, we have a more transient expression in the acute healing group, which is indicated by the orange line here, as compared to the impaired healing group, which is the blue line. And this again shows that these cytokines and these markers are regulated very tightly in the acute healing group, while the levels are kind of fixed and they're not allowing the wound to escape the inflammatory phase in the impaired healing group. Importantly, we're able, without any prior information, with unsupervised learning, to stratify our groups. So this is hierarchical clustering across the different time points. And we can see that our control group, the acute healing group, is clustered together, while the impaired healing group is clustered together. And of course, this is driven by the first time point in most cases. So in summary, our setup, the 100 samples per day SureQuant method, was shown to be very robust with very high sensitivity and throughput. It was very good, and it behaved very well for our data set, which is kind of a big data set in terms of samples, and we had very high data completeness. Of course, there are some limitations. You have to spike in the heavy peptides, so that might be an extra normalization step you have to take into account, sensitivity and triggering of the scans. And the SureQuant methods are require some sophisticated assay optimization. At least their survey runs and preparation need to be more heavily optimized. So this assay would be more suited for methods and workflows that will run over time. So to close, I'd like to thank everyone that worked in this study, mainly Louise, Katrin, and Alexander, who helped with the sample preparation. The DU proteomics core were very helpful in the analysis of the samples, Simonas and Erwin, and of course Hartmann and Professor Hans Mola for providing the samples, our funding bodies, and thank you for your attention. Happy to take questions.